For our first conversation, we're lucky to have two CRISPR innovators here to discuss the potential for gene editing techniques to transform care, as well as the big challenges that surround it. Please join me in welcoming Dr. Feng Shang, an investigator at the McGovern Institute for Brain Research at MIT, as well as a core member of the Broad Institute of MIT and Harvard. Dr. Shang has helped pioneer the development of the genome editing tool CRISPR-Cas9. Dr. Shang and his colleagues continue to refine and improve CRISPR technology and explore how gene editing tools might be used to diagnose and treat disease. His lab tools are being used in the fields of immunology, brain science, and cancer biology. Welcome, Dr. Feng Shang. Next, I would like to welcome John Evans, CEO of Beam Therapeutics, a company co-founded by Dr. Shang that uses CRISPR technology and is working toward creating cures for serious diseases. John has been at the helm of Beam Therapeutics since 2017 and previously held senior titles at Agios Pharmaceuticals, where he co-led the company's expansion into rare genetic diseases. An experienced drug developer and company leader, John has worked at Infinity Pharmaceuticals, McKinsey & Company, and MedImmune. Welcome, John Evans. And here to lead the discussion is Nancy Shute, Editor-in-Chief of Science News. An accomplished journalist, Nancy was previously an editor on the science desk at NPR, where you may have heard her coverage in All Things Considered and Morning Edition. She is a past president of the National Association of Science Writers and a former writer and editor at US News. Nancy Shute, everyone.
Hello. I'm thrilled to be here today, especially back with my wonderful colleagues at US News from US News and World Report, and really excited to be here for a conversation with Feng and John. And Feng and I were talking backstage about CRISPR. You know, we talk all the time about this new technology, and it's new and it's so exciting, because it really has been in less than 10 years that this has been developed as a powerful tool for innovation and now actually clinical therapies. But as Feng mentioned, CRISPR has actually been around for probably millennia, and it's even probably in your breakfast. Can you explain about that? How could CRISPR be in our breakfast?
Well, thank you. First of all, it's really great to be here, and thank you for the opportunity to talk. And it's really nice to be here with John. So CRISPR is a bacterial immune system, and some of you who have had yogurt this morning probably ate some of the bacteria where people began to understand how CRISPR works. So it turns out that CRISPR allows the bacteria, very much like our own immune system, to be able to remember viruses that have infected that bacterial cell. And that way, when the virus encounters a new virus, it already has the immunity and can defend against it. And so people in the dairy industry use this mechanism to make the bacteria that they use to ferment yogurt resistant to viruses. And so when people are growing 10,000 liter vats of yogurt, they don't want the whole thing to spoil. And so they CRISPRize the bacteria so that the production process is more reliable. And so nowadays, 100% of yogurt that's on the store shelves have been CRISPRized, and this is something that all done using a natural bacterial immune process.
That's really amazing. Tell me how you first heard about CRISPR as a process and started thinking that this could be a tool that you could use as a scientist.
So I've been working on this general question of genome editing for a while. And genome editing is about the ability to go into the DNA of our cells, find the right spot, and then be able to make a precise change. Our genome is 3 billion letters long, and so it's a very, very long string. And so to home in and find that exact place in that 3 billion letters is pretty challenging. If the 3 billion letters is in a word processor, it's very easy. You open up a search function. But but the search function sort of in the biochemical sense inside of a cell is much more difficult to develop. And so several different technologies have been developed to make this search function possible. You may have heard of things like zinc finger proteins or talons, and these are some of the previous iterations of the technology that allows scientists to program a protein that they can put into a cell, and then that protein can search along this 3 billion letter string to find the right place. So I was working on this and realized that programming these proteins is really difficult. And one day I was in a seminar and I heard a scientist mention the word CRISPR. The name sounded really cool, so I was curious. And when I went online to look up what CRISPR is, I realized that this is a much easier way to program a protein to find a spot in the genome. And so since then I began to work on it. And nowadays CRISPR is being used as a very easy and efficient way to target different places in the genome, and it's also being used to direct a lot of interesting functionalities so that we can not only do genome editing, but many other things in our cells.
Yeah, and John, how about you? When did you first run into CRISPR?
Yeah, so I don't come at it from the same perspective, but I come at it more from the drug developer side of things. So most recently I was at another company called Agios working on pills, traditional small molecule drugs, but in a paradigm called precision medicine. So we were really targeting patients based on their exact genotype. We knew the mutation they had. We had a drug that could turn it off. That allowed us to really test things early, get an early signal, and then get the drugs approved very quickly. And those are now on the market for acute leukemia. So that was a transformative experience for me. But I was looking for something more powerful and really more of a platform so that if we could do this once, we could do it many times. And that was when I learned about CRISPR and specifically some of the forms of CRISPR we're using at Beam called base editing, which is a new version of it. And that power that Feng mentioned of the ability to take it and target a spot in the genome with becoming a potential medicine, and then making a very simple change and suddenly it'll go to a different spot. Now you have a completely new medicine. Mm-hmm. And now you can do it again and again and again. So the power of that platform was really amazing to me. And it really also struck me as sort of the ultimate precision medicine, where we would go in and find a patient who had exactly one misspelling in their gene and try to turn it back to normal. And that could be a real game changer in medicine. And so in 2017, I switched over to the CRISPR field completely. I've been working with Feng and some of our other co-founders, David Liu and Keith Jung, ever since.
Okay, I want to hear more about base editing and what you're doing with Beam, but let's take a little bit more time to talk about, make sure that people know what kind of gene editing we're talking about. We're not talking about editing something that's hereditary, which is something that's extremely controversial. Can you differentiate that for us?
Yes, maybe I can go first. So everything we're doing with CRISPR today is medical. We're trying to treat people who are sick. And so that is about treating adults, maybe treating children who have a disease, and we're going to try to cure them of that disease. And the amazing thing is we really are talking about the potential for lifelong cures. This could be one-time therapy, and you don't have the disease anymore. So that is a lot of work. That would be an amazing outcome. So we need to be 100% focused on that and not on applying this to anything that could be hereditary or doing things in embryos or the germline, which does get discussed. It's an important discussion, but it's not where we're focused.
Yeah, and I think because CRISPR is so easy to do, it's almost scary how easy to do, and I think that was really forcefully driven home with the revelation that a Chinese scientist has claimed to have CRISPR'd the hereditary genomes of two children. And I think that happened much sooner than anybody really expected, although the ease of it, I guess, would have suspected. I mean, where do we go from here now that we know that there are already people doing that?
Right, so 1 year ago, last November, I think the whole world was shocked seeing in the news that a scientist in China had created 2 CRISPRized or genome-edited baby girls in China. And everyone was shocked, and in fact, there were many different reasons why that scientist shouldn't have moved forward with what he was doing. Since then, a lot of international efforts have convened. The national academies from many countries around the world have established a working group to figure out the most prudent path forward. And also the WHO has a dedicated commission to figure out, we as a society, what we want to do. Now, genome editing at a germline level for many reasons we shouldn't do, both because of ethics, but also because technologically we're not ready to do. And because of that, we really need to convene as a society to have widespread discussions and figure out what does the society want to do with this. Eventually, the technology will become ready, and so people will be more and more tempted to apply this in the germline setting. But without societal consensus, I think it will be very dangerous to move forward too quickly. And so that is the conversation that's going on now.
Yeah, and I think it was the fact that you mentioned it's too early. This is an amazing technology, but it does make mistakes. And as scientists do more work, you and your colleagues, we realize it's not perfect. And so I know that a lot of people are working on trying to come up with solutions to that. Tell me what you're trying to do at Beam.
Absolutely. So, yeah, the technology is moving very quickly. So important to remember that we also, are part of the gene therapy field, right? So gene therapy was the first wave, and we were talking before about how gene therapy always seemed to be the medicine of the future, but maybe it never quite arrived. Well, it's here, and we're getting drugs approved now. So gene editing is next, and gene editing is now reaching the clinic, and that search function that Feng mentioned is so powerful to be able to target any spot in the genome. Where Beam is going is even a next generation beyond that, because once you get to that spot in the genome, what happens next? So the traditional CRISPR is like a scissors, so it'll cut the genome at that spot. And that's very powerful, and it's useful for some applications, but we would actually like to exert more control over what you do once you get to that spot in the genome. And so what Beam does is something called base editing, where we can actually land at that spot and then make a single letter change in that gene without needing to cut it, and then let go, and you've again permanently changed the genome. So sort of the ultimate, again, precision edit. And that's going to be really useful for the fact that, for instance, the most common class of genetic changes in disease are point mutations. So a single letter misspelling in a gene. So we would like to go in there and turn that back to normal. So something like sickle cell anemia, everybody who has that disease has that same single base misspelling. That would be something that we would be really interested in targeting.
What are a few other single point mutations you're looking at as possible?
Yep, another famous one is called alpha-1 antitrypsin deficiency. So that's a liver and lung disease. Again, every patient has the exact same single letter misspelling, and we would be looking to make that exactly back to normal.
This was a very big year for CRISPR therapies because we're seeing the first clinical trials with CRISPR. Can you two talk a little bit about what you see happening there?
Yeah, amazing progress by the field. So it's unbelievable that I think CRISPR as a therapeutic application wasn't even described until about 6 or 7 years ago,
crazy?
right?
Yeah.
And here we are in the clinic, and already there's newer forms of technology coming along. So the companies that have been pioneering this field have done an amazing job. It's easier to forget, but this is really going to be an entirely new class of medicine. So all of the manufacturing and how you supply it and the regulatory package, how you measure specificity, has to be invented from scratch. And there's been a tremendous amount of progress already, enough that it's now deemed fit for human testing. And so those trials are now ongoing. And then I think we will learn a lot from those first few trials over what the pathway forward looks like.
So we're seeing a trial for sickle cell, several cancers. What other clinical trials are in the works or have?
There's an eye retinal blindness from Editas as well. So really a wide variety of organs, disease types, and applications are already being tested.
One of the interesting twists to me is whether you're going to edit the genes outside the body or inside the body. And it seems like it'll be a lot easier to do outside, but I would love to hear your thoughts on that, Feng.
Yeah, so there are two ways to go about doing CRISPR gene therapy. There's what's called ex vivo, which means for diseases affected by cells that you can take out of the body, like in the blood system, or for treating cancer by changing the immune cells. You can take the patient cells out in a petri dish, you can modify it, make sure it's all done properly, and then transplant back into the patient. That's called ex vivo therapy. The other kind is called in vivo genome editing, and that's for tissues that you can't take out of the body. You can't take someone's brain out and then expect to put it back successfully. So for that, the approach is to directly deliver CRISPR into the patient, whether it's in the liver or in the brain or muscle, and that's in vivo. And that's usually done now using viral vectors. So the same vehicles that people in gene therapy use, and adapting that to deliver the CRISPR system into the affected tissues.
I think that's what they're using for the retinal disease trial, right? It's in vivo, because again, you can't take your retina out and you can't do a retina transplant, so there has to be a way to get the CRISPR tools inside the eye, which is remarkable. Yeah. And it sounds like at this point we haven't seen, there's been no like, oh my goodness, we've cured this disease. It's more like early indications are that these have not caused a danger to patients, and therefore we can move forward with testing in more patients. Does that sound about right?
Yep, I think that's right.
All right. And now, as you're thinking about this as trying to scale up your technology and potential therapies, as you mentioned earlier, this is something that could be incredibly disruptive to healthcare delivery because you might be able to cure patients. How do you what would you like to tell healthcare professionals about what they should be thinking of, and both in terms of a timeline for these things potentially reaching the clinic, and also how it might change healthcare delivery?
Yeah, that's a great question. So our mission statement is to deliver lifelong cures to patients suffering from serious diseases. So that is an amazing concept that we're even having that conversation. And so I think in terms of timeline, back to the idea that these are precision medicines, we're treating patients who have the disease. We know who to treat. So if you see a signal, you're going to sit with the FDA or a regulatory body and say, how fast can we move this to approval? Because patients are waiting. And you don't have to take 8 to 10 years to get that kind of a drug approved. It can be 4 to 5 years. So I think it's possible that these will move fairly quickly, and that's been happening already in the gene therapy field. So although it is early, and although it's not here today, I do think for those in this audience, you should be thinking ahead for what it will look like as these sorts of therapies do come online over the course of the next decade. And they will be disruptive. So these, if you think about it, this is a patient who, without the therapy, is going to be very sick, have incredible morbidities forever. And they're going to be constant users of the hospital system and of care, not to mention therapeutics. And here we have the potential to, with a one-time therapy, reduce that burden or even eliminate it and cure that patient. So that's going to be a totally different paradigm for treatment. And I think ultimately it's going to provide a lot of value actually back to the system if you think about all those years that you can potentially be avoiding the disease for that one-time therapy. Nonetheless, to do that, those therapies are going to be premium priced. And so getting that balance right is going to be really difficult. And so I think, again, the folks in this audience, we can work together on that. There's already a lot of innovation happening in the gene therapies as they're launching. So we're watching that very closely because the same sorts of lessons would apply for gene editing as well.
And Feng, you mentioned an interesting way to think about it. I guess if I was an oncologist, I might be thinking, am I going to be out of work in 10 years? But you said if you think of it as genome surgery, it is a complex medical care that people will have time to learn how to live. Tell me how you think that might work.
Surgery, when you think about it, is about making an incision and going in and repairing things in the body. But as we start to understand the biological mechanism, like the basic biology of how disease happens in our cells, then the idea is to use these genome editing tools, go into the DNA, make the right incisions and make the right changes directly in the DNA. Now that will require us to provide the tools and the training for a new generation of molecular or genome surgeons, but I think that's a really exciting future for medicine. It will complement the existing medical approaches and surgical techniques, but augmenting that toolbox so that we can have even more available solutions for patients who are affected by these devastating diseases.
One of the big changes, go ahead, John.
Well, I was just going to build on that just to say that I think that paradigm is going to become really important given that some diseases like sickle cell anemia or alpha-1 antitrypsin deficiency, everyone has the same mutation. So just one product potentially treats all those patients. There's lots of other diseases of patients who need care where the mutations will be very different. And so that's going to require something you were saying before, a more bespoke, personalized therapy. And that's where I think it does merge back in with the provider setting to do that in a more increasingly customized way, which is really interesting.
Actually, to add to that, in order to enable that personalized approach, regulatory processes need to adapt as well. You don't want to have to run a new clinical trial for a new mutation, especially those only affect 4 or 5 people in the world, the tail distribution of rare genetic diseases is very long. And so as we started to think about how to deliver this technology to all those rare disease patients, we really have to think about how to go about testing the safety and efficacy of these new drugs.
Yeah, and with personalized medicine, because it is very individualized, it's also often very, very expensive. How do you see the CRISPR therapies fitting into those profiles in terms of cost?
Well, as I said before, I think they will be similar to the gene therapies that we've already discussed, and that's just because of the novelty and the expense of developing them in the first place, and then the fact that there's not a lifetime of revenue from a given patient. It's really just a one interaction. But again, I'd come back to value. I really believe that if you can treat a patient in the prime of their lives, if you can treat a child who has a productive life ahead of them, I think the health economic analysis will actually be positive, so long as we can find a way to structure the payment in a way that works for the system. And I think that's been the conundrum.
So when you're talking to healthcare professionals and executives like the folks in this room, we already talked about some things they should be aware of, including timeline. I'm thinking too in terms of what fields of medicine do you think will be most affected by CRISPR therapies? Any guesses on that?
Well, I think I'll start.
Yeah.
I think what CRISPR or gene therapy is making possible is for doctors to be able to treat many more diseases that don't have any treatment options today. And so in many ways, you can think of it as expanding the number of treatment strategies and expanding the number of patients that we can actually effectively care for. So it's not replacing existing medicine, but it's really making us more capable, do more.
Yeah, I would agree with that. I think of the future, and small molecules will still have a place. I'm sure chemo, unfortunately, will still have a place, antibodies and proteins. But I think the question now is just what share of the pie will be genetic therapies, and I think it'll be a significant share and one that grows. I also think that as the technology matures, we will go from treating purely very severe monogenic disorders, which is where we're starting, to treating increasingly common conditions using these tools. And I think that is possible. New paradigms like preventing disease, where you edit a tissue to try to cause that tissue not to develop the disease later, building on clinical genetics and things that we know about populations who don't get disease. So I think there's a lot of different applications that over time will become plausible as we reduce this to practice. But again, initially, really only about severe and genetic disorders.
And that, in the long run, it will open up more ethical questions too. We talked about earlier, modifying the germline, so changing hereditary things. But genetic enhancement in the adult human body is another thing that is actually quite challenging. And so ethically, I think another important conversation to have is how far do we want to allow this technology to be applied? In the future, would you want to be able to preemptively change certain things to make someone even smarter or even more capable, and how that will affect the inequality that we already face in the society? Those are the big questions that we'll eventually have to deal with.
That's really interesting, because I think so much of our focus in the last year has been on the ethics of germline editing. But you're right, I could gene edit, I could CRISPR myself if I wanted to, and it would be so much more effective than plastic surgery. Or my children, and then again, you get into some profound ethical questions.
Yeah. The only thing I love that line of thinking. It is important to remember there's an asymmetry in our information, right? So in disease, we know perfectly what causes the disease, because you have a healthy genome, and now you look at the patient who has a disease, you know the one thing that's changed, it's that mutation. So if you fix that mutation, you know you'll cure the disease. Things like intelligence, strength, being a well-behaved child, these are not things that we know the gene for, and probably it's not one gene anyway. It's probably 1,000. So I think that all of that is probably not going to arrive quite as quickly. But
okay, we've got just a few more minutes. What is your big challenge for the next year, Feng? What are you working on now that you want to?
I think one of the biggest problems facing not only gene editing, but gene therapy as a whole is delivery. The reason that you see the existing approved gene therapy or many of the clinical trials in gene editing, the reason you see that they're focused on the eye, the liver, and also cells in the blood is because those are the places where we have reasonable ways to deliver into. Vast majority of the other parts of the body, the heart, all of the muscle cells or the bones and even the kidney are much more difficult to be able to deliver things into. So unless we solve this delivery challenge, we wouldn't be able to open up this wide array of possibilities, and that's one of the things that we're working on now.
And John, what's on your list for this year?
We're growing, and so it's really execution, actually. We've got the technology of base editing in hand. We've applied it to many of these diseases, and it looks really good in that cell stage. So now, over the course of the next year or so, it's really about moving it forward. Again, following on some of the tracks that have been laid by others, who are working on this field already, so that we can begin clinical testing. And I think, so for us, it's really about growing the team, thinking about all the different disciplines that need to come together to make what is a very complicated molecule become a medicine. And that means manufacturing, it means specificity, it means regulatory, clinical, preclinical testing. But all of that is really in motion. So we're very, very excited about the future.
So it sounds like it's going to be a very big year in another very big year in CRISPR in not even a decade that's been quite extraordinary in science.
My favorite quote is Bill Gates, where he said, you can always accomplish much less than you think in a year, but much more than you think in a decade. And I think if you look at the CRISPR field, you can see that in action.
I agree.
Yep. All right. Well, thank you both. It's been a fantastic conversation, and I really appreciate you coming and joining us today. It's been outstanding.